Welcome to Let's Get to the Marks. This is an AQA A-Level Biology Paper 3, and we're looking at the essays. And this is video number 4. Check out my other videos, and it's on ions. Now, there are lots and lots of different essay titles you can get in AQA. You get the choice of 2 at the end of the paper 3. And today, I'm going to write an essay on the importance of ions in biology. But this style of essay has come up in lots of different forms. For example, another one over here, the importance of ions in metabolic processes. You just need to adapt it a little. And you can really, if you follow this, you'll be able to do any essay on ions. Okay, so once again, the title's write an essay on the importance of ions in biology. So the first thing you want to do is brainstorm out. Can you think of 5 topics on this title? That's very, very important. So you get your 2 essay titles and you need to spend 2 minutes with the one you choose, making sure you can pick 5 topics. You need to be able to cover AO1, which is scientific knowledge, and be able to understand how to do the AO2, which is explain the importance or the application of the content. Now, as long as you can do the scientific content, the application of the content should be fairly easy. You just need to say why it's important, what you're talking about, and how it applies. So here's my plan on ions. Now, there are many, many different topics you can talk about. I'm going with the nitrogen cycle, photosynthesis, respiration, the nervous system, followed by muscle contraction, and beyond the specification if you're looking to get beyond 20 out of 25 marks. But of course, you could drop one of these 5 topics if you have a real solid beyond the specification. That will be your 5th paragraph or 5th topic. So you don't need to have 6 like I've got here. I'd probably, if I've only got 40 minutes to write the essay, I'd probably choose to drop one of these if I'm really solid on my beyond the specification. If I'm a bit doubtful, like I can only do 2 or 3 sentences on the beyond the specification, then I might want to add in that extra topic just to make sure I get up towards those 20 marks if I'm a bit worried my beyond the specification's not good enough. Now, do you need an introduction? Not really, but sometimes people like to have an introduction. It helps them flow into their essay. So to focus on your introduction, think about the title and just define what it is. So this essay is on ions. So I've just started with inorganic ions are charged particles, e.g. sodium ions are positively charged, phosphate ions are negatively charged. In this essay, I'll describe their importance to living things, including plants, animals, and bacteria. Okay, so my first topic's the nitrogen cycle. I've got a little subheading here. You don't need to put that in your essay. Don't put subheadings. Nitrogen in the atmosphere is fixed by nitrogen-fixing bacteria that live in the roots of leguminous plants. They reduce nitrogen to ammonium ions using ATP and hydrogen ions from reduced NAD. The ammonium ions are then oxidized by nitrifying bacteria such as nitrosomonas. That's a bit beyond the spec, but it's nice to put it if you can remember. If you can remember, just write nitrifying bacteria. If you can't, you don't want to contradict or say something incorrect. So nitrifying bacteria like nitrosomonas turn the ammonium into nitrate ions. The nitrate ions are toxic to plants, so other nitrifying bacteria need to turn those, nitrobacter as an example, or oxidize those nitrate ions into nitrate ions by adding the extra oxygen. The nitrate ions can then be absorbed into root hair cells by diffusion or active transport. Why is all of this important, talking about nitrate ions in the soil? These nitrate ions are important because they increase the nitrogen content of the soil, enabling plants to make ATP, because that contains nitrogen atoms, and it's the energy transfer molecule, and to be able to make DNA the universal code for life. You can go into a bit more depth about why ATP is important for muscle contraction, etc., or why DNA is important as well if you want to, but you might be going a little bit off topic there and you want to stick within your time limit. The nitrate ions can also be used to make amino acids, okay, and therefore proteins. And why are proteins important? Well, proteins can be used to make hormones like IAA, which are used in the phototropic responses, and to make enzymes such as ribisco, which we know is needed in the light independent reaction or the Calvin cycle of photosynthesis. Now, why I've kind of finished this nitrogen cycle bit there with the word photosynthesis is because that's going to be my link to the next topic. There's a bit of controversy, or there's a little bit of ambiguity as to whether you need to link the paragraphs. When the new spec came out in like 2015, AQA said, yeah, you need to link the paragraphs, but they've kind of wound back on that now and said they're not so interested in you linking the paragraphs. Now, I've highlighted the word important here. This is kind of like this purple bit here is kind of my AO2, and I like to keep using the word important because they always put the word important in the essay title. So just remind the person reading your essay that you know why these things are important. And notice I've highlighted the word photosynthesis, and that's because my next paragraph is gonna go into photosynthesis. So I've started here, probably not a very good place to start, but plants can use active transport of nitrate ions. So I'm kind of linking it to what I've been talking about to lower the water potential in their root hair cells and therefore increase the water uptake by osmosis. I should have said to increase the water potential gradient, but there we go. Water is needed in photosynthesis, and now I go into during photolysis, water is split to release hydrogen ions. Remember, we want to highlight the ions and electrons. The hydrogen ions I could put here are important, but I've put are used in chemiosmosis, where the kinetic energy of their passage through ATPase or ATP synthase in the thylakoid membrane is used to join ADP and PI. And if I want, I could say inorganic phosphate ions and produces ATP. And together with electrons, they reduce NADP. This is important because the reduced NADP enters the stroma in the Calvin cycle, and using the electrons and hydrogen ions and energy from ATP, it's going to reduce glycerate 3 phosphate to triose phosphate, which goes on to form those organic molecules like lipids, glucose, proteins, cellulose, etc. Now, I could stress here again, the purple is my kind of why is it important, my AO2. So without the hydrogen ions released in photolysis, the plant couldn't produce glucose. This is therefore telling us. So it wouldn't be able to do respiration and produce ATP. Glucose, lipids, proteins, and other biological molecules produced are important for the plant's survival. I could go into the fact what proteins are used for, i.e., to make enzymes. I could talk about the lipids and the waxy cuticle if I wanted to, etc. You could sort of really stress why without the hydrogen ions a plant would be screwed basically. And also I'm linking to my next paragraph. I'm saying um, this is also important in the ecosystem or the food chain because animals consume plants to get those biological molecules. And now that links into respiration. The glucose consumed by animals is used in respiration, in which ions play a vital role in each stage of respiration. Phosphate ions released from the hydrolysis of ATP are used to phosphorylate glucose in glycolysis in the cytoplasm. Here I've just tried to put some words in bold just to give you an idea of these paragraphs are really hitting the content. And respiration and photosynthesis are great for hitting detailed A-level knowledge because there's so many key terms. So I really love those if I can work them into any essay. The pyruvate formed enters the mitochondria in aerobic respiration and transfers its hydrogen ions to the coenzymes NAD and FAD in the Krebs cycle. These coenzymes release the hydrogen ions in oxidative phosphorylation in the cristae. The hydrogen ions are actively pumped into the intermembrane space of the cristae. They then move down an electrochemical gradient by chemiosmosis through ATP synthase and produce ATP. Again, you can go into a little bit more about why this is important. So I've got here in purple the hydrogen ions, the ATP, muscle contraction, and I've tried to end on nerve impulse because that's my next paragraph. So then I go into nerve impulse transmission and essentially talk about in nerve cells, ATP to ADP and PI provides energy for that should be sodium potassium pump, I guess it doesn't matter, to actively transport 3 sodium ions out and 2 potassium ions into the axon of the neuron. As the membrane's sodium ion channels are closed, it has low permeability to sodium ions, and therefore it's important as this enables us to have a negative resting potential across the membrane. So it's minus 70 millivolts inside the axon relative to the external environment. This transport of ions is really important as when the sodium gates open, we've got this influx down an electrochemical gradient of sodium ions into the axon, and that causes the generator potential. So without that pumping out of the ions, we wouldn't get a generator potential when the gates open. And if it crosses the threshold, the membrane depolarizes, plus 30 millivolts, and an AP will form. During repolarization, the potassium gates are open and the potassium ions leave the cell. This has to happen for repolarization, otherwise the nerve couldn't recover. As the gates are so to close, the axon becomes hyperpolarized, which allows for the refractory period. So that's a bit of AO2, like what is it good for? You know, we need that refractory period so that we get discrete signals, which I didn't mention there and I could have done. It is these waves of depolarization caused by movement of ions that allow animals to respond quickly and effectively to both the external and internal stimuli, allowing rapid movement, coordination, and homeostasis, all increasing the chances of survival in the environment. Now, I'm not going to go into a fifth topic here, but that's that's a lot of content I've covered there, and you'd definitely be scoring the content marks. I probably could have stressed a bit more AO2, but here's some other ideas for content. Um, I don't really like this topic, but you could talk about ventilation. We could talk about oxygen coming into the lungs. We could talk about Fe3 plus ions, okay, and their role in heme groups within the hemoglobin. And we could talk about that. We could talk about the chloride shift, carbonic acid, and all that business if we want with the blood. I don't quite like that topic for essays. What I could really go into is this one. Calcium ions are essential to muscle contraction. That would follow beautifully after nerve impulses, right? And I could talk about the calcium ions joining to the troponin, changing the shape and making the tropomyosin move out, freeing the actin myosin binding sites on the actin filament where the myosin heads go. This enables the formation of cross bridges. So without this, we wouldn't be able to get cross bridges. Also, the calcium ions activate ATPase, which hydrolyzes the ATP to ADP and inorganic phosphate ions, releasing the energy for the power stroke. So without those calcium ions, ATPase wouldn't get activated and the muscles wouldn't be able to contract. They wouldn't be able to do the power stroke. So that's a topic we could really get into in quite a bit more detail. You've also got transmission across the synaptic cleft. Again, the calcium ions with the channels opening. That's another really good topic. You could go into some detail there. So there's loads and loads of topics you could get into as a fifth topic if you've got time. You should be spending 40 minutes, and I reckon at least 3 to 4 minutes planning out so you get the best essay. Okay, because you might not like one essay title and you might prefer the other, but if you sit there and try to quickly write out the topics that you could talk about, you might find the one that you think is a bad essay title is actually the good one. So it all depends on which one you can come up with at least 5 topics. So moving on swiftly, we're all interested in the beyond the specification. And also, what I want to say is all of these topics I've talked about, you want your rock solid topics like photosynthesis. Photosynthesis can come up in proteins, where you talk about the ETC and the photosystems. Photosynthesis can come up in membranes. Photosynthesis can come up in movement within the cell. You can talk about the electrons moving. It can come up in essays on carbon dioxide with Rubisco and carbon fixation. So one good thing is to go through and each time you're revising a topic, think how many different essays could I get this topic into? And you should be doing that when you're reading your notes and revising for paper 1 and 2 as well. Just be thinking, where could I stick this bit of info in an essay? You'll find quite a lot of topics, again, like respiration. They involve facilitated diffusion. They might involve oxidation reduction, so they could come up in redox, electron transport, carbon dioxide, cell membranes and membranes within cells. There's like so many different places, and of course they everything in biology fits into proteins in some way, like hormones, enzymes, carriers, protein transport channels, co-transport channels. So everything can work into proteins, and that's what's great about that. Right, here's my beyond the specification. Doo, 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 doo. Okay, we got Vibrio cholera. Now, this is from the old spec. If you've got an old AQA book, you can look online, an old AQA specification. You'll find this is there. And basically, it's a species of bacteria which is spread through contaminated water. Check out the grade D GCSE art. It uses a corkscrew motion of its flagella to burrow through the mucus lining of epithelial cells of the small intestine. So that's going to get you marks. It produces a toxin that's made of 2 parts. The first part has a complementary shape specific to carbohydrate receptors that are only found on the small intestine epithelial cell membrane, hence why it only affects the epithelial cells in the small intestine and it doesn't attack the lungs or something like that. So, one of its bits binds, one of its proteins, 3D structure, binds to that membrane and it causes chloride ion channels to open and chloride ions to move by facilitated diffusion into the lumen of the small intestine. What's that going to do if you pile loads of ions into the small intestine in the lumen? It's going to lower the water potential in the small intestine. And also, it's going to raise the water potential in the epithelial cells because there's less chloride ions now in the epithelial cells, which means water is going to move by osmosis into the lumen and water is going to move, and more chloride ions are going to move down in a diffusion gradient into the epithelial cells from the blood and surrounding tissues. So loads more ions are going to move in there by diffusion, and then water's going to follow behind by osmosis. So it sets up a chain of ions moving, followed by water moving by osmosis. And the result of this is dehydration and diarrhea because you've got all that watery feces going on in your intestine. and the cells are losing loads and loads of water. It can also be treated by oral rehydration therapy. There's a whole load of that on the old spec. You can go and look at some old CGPs. You might get them on eBay on the cheap, and you'll find bits of content that were in the spec before 2015 that aren't anymore. And you know they're at A-level standard because they're from old A-level specs. So that's what I've done here. And this is a great piece of information, this cholera one, because again, it can be used in for ions. It can be used for membranes. It can be used for disease, bacteria, the effect of microorganisms or importance of microorganisms in biology. There's all sorts of things this can be used for. All right. So that is about it. Make sure that you subscribe, like the channel, and check out all my other essay videos that I've got going on. You might want to add a little conclusion. You could put something like, once again, here we can see the importance of ions in biology, blah, 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 blah. You can make a little conclusion if you want. Here are the topics. This essay came up. Here are the topics that I didn't cover in red. So phosphate in the structure of phospholipids, and the importance of ions in DNA. Well, you've got the phosphate ion, which has a negative charge. That's part of nucleotides and part of DNA. I didn't really want to talk about that. Water potential osmosis, chloride ions, and cholera. You can tell this is from the old spec because cholera is no longer on that. So BS means beyond the specification. It doesn't stand for, that rude word which might describe my videos. Co-transport involving sodium ions. That's a brilliant one, and I didn't cover that in this essay, but you can talk about the co-transport mechanism. It's a bit similar to the cholera mechanism there. Hemoglobin and iron I didn't talk about, and I steer away from that one. Passage of water through plants, symplast, and root pressure. That could be another beyond the specification one. I didn't talk about that, but they took that information out of this spec that you're currently doing. So yeah, I did cover this, protons or hydrogen ions in photosynthesis. Yeah, we did that. And I did the reduced NAD and FAD and phosphorylated intermediates. And I did mention glycolysis, but I didn't really talk about glycolysis into lactate and how ions play a role in that. And I didn't do the use of NPK fertilizers, which I could have worked into my nitrogen cycle if I wanted, but I did talk about the nitrogen cycle. And I didn't do chemoreceptors, heart rate, and Pacinian function. But I did talk about nerve impulses, but not really at the synapse. And calcium ions and muscle contraction, I always think that's just a great one to do. It's really easy to remember all the detail for that. Genetic fingerprinting and electrophoresis, I probably wouldn't do. And you might be wondering how ions are involved. Well, you've got to think the phosphate ion on the DNA gives it a negative charge, allows the DNA to separate your different DNA fragments to separate as they move towards the positive electrode through the gel in electrophoresis. So if you're an electrophoresis nut, you could really get into that if you wanted. All right, guys, that's all for me. Don't forget, let's get to the marks.
You're still here? It's over.